Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com Inside Scientific and Bio-Rad webinar titled Fast Forward Your Custom Antibody Generation with 4 Bio-Rad Services. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Renee Brissette, who will discuss advancements in bioanalytical antibody development and highlight strategies for ensuring high-quality, reproducible results. And with that, I'd like to welcome Dr. Renee Brissette. So, Renee, the floor is yours whenever you're ready.
Hello, everyone. I am a technical sales specialist for Bio-Rad custom antibodies. Today, I will overview 4 Bio-Rad services to fast forward your custom antibody generation. Today's agenda will include the following topics. First, I will overview the UCAL Platinum Library, the phage display antibody library we use in our in vitro antibody generation platform, discussing briefly its construction and the proven success of this library. Next, because customers come to us frequently with the same 4 requests for their custom antibody generation, namely speed, high specificity, high affinity, and quick format and labeling options after generation, I will discuss our 4 services to deliver these requests. Lastly, I will give you some examples of high specificity antibodies we have generated, as well as a brief summary. The UCAL Platinum Library. We have a very successful proven library called UCAL Platinum, which stands for Human Combinatorial Antibody Library. We have generated more than 58,000 antibodies for our customers to date. It's a library of antibody genes. The genes encode Fab fragments shown in the upper middle panel. This is a part of the antibody that contains the variable regions, and it's these variable regions that contain the antigen binding site or the paratopes, as shown in the upper left panel. It's a fully synthetic and fully human library. It is a very large library composed of 45 billion unique antibodies, one of the largest high-quality antibody libraries worldwide. The fully synthetic framework genes shown in the upper right panel are based on consensus sequences of human antibody genes and cover the diversity of the human antibody germ lines. The hypervariable regions of the antibodies are also known as the CDRs, or complementarity determining regions, are generated by a process called trinucleotide mutagenesis and inserted by pre-built cassettes. These features lead to the enormous diversity we see in the UCAL Platinum Library and guarantees the successful generation of high-quality antibodies. Further, Bio-Rad has its proprietary cis-display technology incorporated into the construct. Fab fragments are linked to phage particles by a disulfide bond rather than a peptide bond, as shown in the upper right. This allows elution of phage with reducing agents during antibody selection. This ensures that all phage displaying specific binders are eluted from the antigen, including ones displaying very high affinity antibodies, which cannot be easily retrieved using standard elution conditions such as acid elution. Our panning service delivers speed. The antibody generation process takes as little as 8 weeks. This is our workflow as a schematic diagram. In the upper left corner, the tube is the UCAL Platinum Library, and the orange panel is the customer supplied antigen. 3 rounds of panning are carried out, and our guided selection core technology, which I will discuss next, can be applied in all 3 rounds. The phagement DNA output is recloned into an expression vector, transformed into bacteria, and single colonies are picked. Around 384 individual clones are grown in deep well plates, and cell lysates are produced containing the UCAL antibodies. In the screening process, shown in the upper middle panel, lysates are tested for binding to antigens or CRAs, closely related antigens, and hits are identified. At this point in the process, The secondary K-off rate screening can be carried out also, which I will discuss later. Hits are sequenced so that only unique clones are moved forward. The unique hits are expressed and purified, and the purified antibodies undergo quality control. This process is accomplished in just 8 weeks, delivering purified antibodies in the Fab format. And in just 1 additional week, we can generate more format options via our SpyTag technology, which I will discuss later, such as bivalent Fabs and IgG-like molecules, all with or without labeling. We call these coupled antibodies trailblazer antibodies. What this means to you is that we can deliver a set of antibodies, for example, 10 antibodies. Then we can quickly and cost-effectively generate multiple formats for each antibody. For example, 2 additional formats for each antibody. We would ship to you not 10, but 30 different antibodies that you can test in your applications. And if, however, you would like a full native antibody without tags, we can generate that format also in an additional 8 weeks, as shown in the bottom panel. And we can add more speed by accomplishing this antibody generation process in only 4 weeks. That's half the time. Here, the panning can start 2 working days after receipt of the antigens. The product duration is 4 weeks from start to finish, or 5 weeks if SpyTag coupling is included. Our guided selection core technology service delivers high specificity. In this first selection example, we achieve epitope specificity. Fab antibodies recognize antigen, but not a closely related antigen, the CRA. We black with the CRA in solution the white dots, as shown. Here, the selection is on the immobilized antigen, the blue dots. The antibody phage library is pre-incubated with the CRA or CRAs, and then this mix of antibody-carrying phages and the blocking solution is transferred to the immobilized antigen so that there is a competition between the soluble CRA in excess in solution and the immobilized antigen. And then the antibody-carrying phages will bind to epitopes common to both the antigen and the CRA, and should bind more likely to the CRA in solution because simply there is more of it there, and then are washed away. The antibody-carrying phages that specifically bind to the antigen are eluted with reducing agent in the CysDisplay technology, and the enriched phage is amplified and subjected to 2 more rounds of panning. In the second selection example, we achieve cross-specificity. Fab antibodies recognize epitopes present on 2 or 3 antigens, for example, a human and mouse target. We alternate the antigens during the panning. We use the human antigen, for example, in round 1 and 3, the blue dot, and the mouse antigen in round 2, the red dot. Our secondary K-off rate screening service delivers high affinity. As the name indicates, the secondary K-off rate screening comes after the primary screening in the antibody generation process. The screening is a high throughput off-rate ranking process using BLI and the OTTEC instrument, an antibody fab containing crude E. coli lysates. The off-rate was chosen because the off-rate is concentration independent and can be applied to lysates in which the antibody concentration is not known. In this extended antibody screening process, the right side of the figure, clones with the best signal in the primary ELISA are ranked according to their binding strengths, the K-off rate determination. So we then continue with sequencing of the best performing clones from the K-off rate ranking, having the slowest off rates. Selection of antibodies can now occur to both antigen specificity and also binding strengths. Our SpyTag technology service delivers quick formats and labeling options. The basis for SpyTag technology is the formation of a spontaneous isopeptide bond between a lysine and an aspartic acid residue in a domain of the fibronectin binding protein of Streptococcus pyogenes, the organism where it gets its name. Discovery research showed that the immunoglobulin-like collagen adhesion domain of the FBAB protein could be split and the fragments modified to result in a peptide in the middle panel that rapidly formed an amide bond to its protein partner shown in the left panel. The peptide and protein fragments were named SpyTag and SpyCatcher, respectively. The SpyTag can be fused at either terminus or internally. The reaction between SpyTag and SpyCatcher is spontaneous, only needs mixing. It's fast, requiring only minutes. The reaction is robust, occurs with high yield in diverse conditions of pH, temperature, buffer, detergents, and the reaction can occur in cell lysates and in inside of cells, so it is an extremely useful reaction. So we are using this technology in the context of our custom antibodies. We have incorporated the SpyTag at the C-terminus of our monovalent Fab format as shown at the top panel. The SpyTag forms a covalent isopeptide bond to a SpyCatcher protein in the bottom panel, and in this way, the SpyCatcher protein can add a function. And we have a menu of catchers. For example, the SpyCatcher can be site-specifically conjugated to biotin, enzyme, or fluorophore. It can be fused to a second catcher to make a bivalent mini antibody, or SpyCatcher protein can be fused to an IgFc to make a full-length IgG-like molecule. And we have SpyCatcher proteins fused to the Fc domain of, for example, human, mouse, and rabbit. As mentioned, the SpyCatcher protein can add the function of biotin, enzyme, or fluorophore. To do this, we engineered cysteine residues into the SpyCatcher proteins, and we are using malamide chemistry to site-specifically label these domains with biotin, enzyme, or fluorophores as shown by the orange circles. There are a number of advantages to using labeled SpyCatcher proteins as a way of generating labeled antibodies. First, the site-specific nature provides for a fixed degree of labeling and a high batch-to-batch consistency. Another advantage is because the labeling reaction has occurred before the Fab is coupled. We are confident that there has not been any incorporation of the label into the antigen binding site, which could affect binding. And we can make a large batch of the pre-labeled SpyCatcher proteins, and multiple antibodies can be labeled with the same batch of pre-labeled SpyCatcher. And their performance can be compared more accurately because the variability in labeling has been omitted. The advantage to you is that each antibody can be converted to multiple formats in an instant, and you can test all of these in your applications. With QC added for the coupled antibodies, only one more week is added to the antibody generation process, 8 weeks to 9 weeks to receive these coupled antibodies. Lastly, I will discuss examples of high specificity antibodies that we have generated by our guided selection core technology service. We generate anti-idiotypic antibodies, and we are in the position of a market leadership in this space. Anti-id antibodies help preclinical and clinical research. Anti-ids are useful in bioanalytical assays that provide data that is an essential component of clinical trials for therapeutic antibodies and biosimilars. Anti-idiotypic antibodies are directed against the idiotopes of another antibody. The idiotopes are located in the variable region of an antibody and can overlap with the paratope, the antigenic binding site of the antibody, as shown in the circled part of the antibody figure. The anti-id is specific for one particular antibody, and they are inhibitory if they bind the paratope. We use our guided selection core technology service to deliver these highly specific anti-id antibodies, and we use the same workflow as discussed before. For the guided selection strategy in this case, the selection is on the drug in the presence of an isotype match control blocking protein, and in the presence of human serum for the avoidance of matrix effects. Anti-idiotypic antibodies can be used in the PK assay to detect the antibody drug after administration in humans. The challenge is detection of human or humanized drug within a million-fold excess of the patient's antibodies, comparable to finding a needle in a haystack. And the standard assay format today is still the antigen capture, as shown in the left panel. The antigen is immobilized. You come in with the drug and human serum, and the detection is through the FC. There are two challenges here. One, do you have the antigen available? If you're working with TNF alpha, it is easy, but if you're working with a GPCR, it might be really difficult to get the soluble antigen, or it may be extremely expensive. The second challenge is using an anti-FC detection. If you're working in human serum, you will have high background and sensitivity is not really good. Our suggestion for a better assay is the anti-idiotypic bridge. We developed anti-idiotypic antibodies to be used as capture and detection reagents in the right panels. You have a very clean assay, no background, and you don't need an antigen. And these assays are used today in clinical trials to measure PK of therapeutic antibodies. And we even went one step further, and we developed anti-idiotypic antibodies to answer three questions asked for in bioanalytical assays. One, what is the amount of free drug? Two, what is the amount of total drug? And three, what is the amount of bound drug? We call them type 1, 2, and 3. Type 1 binds to the paratope. It is inhibitory. It detects free drug. It's the classical version and relatively easy to get. Type 2 binds outside of the paratope. It is not inhibitory, and it detects total drug, both free and bound. The last, type 3, if you are exact, this is not an anti-idiotypic antibody since it binds to a new epitope formed between the therapeutic antibody and the ligand. It is not inhibitory, and it detects the bound drug exclusively. We use 3 different strategies in our guided selection core service to generate these binding modes. For the type 1 inhibitory binding mode, selection is on the free drug, gold. Blocking is with the isotype match control antibodies, gray, plus human serum. For the type 3 non-inhibitory binding mode, selection is on the drug target complex, gold and red. Blocking is with the isotype match control antibodies in gray, and target antigen in red, plus human serum. Here is an example of all 3 binding modes. Golimumab is an immunosuppressive drug blocking TNF alpha. Type 1 only binds the free drug, left panel. Type 2 binds golimumab in the middle panel in the absence and presence of TNF alpha. And type 3 is just binding golimumab in complex with TNF alpha in the right panel. This is an example of a PK assay bridging format. This is for trastuzumab. In the panel on the right, we have an anti-id capture immobilized. We come in with the drug in human serum, and then we have the anti-id detection. You can see here we are working in 10% serum, and the activity starts in the area of 0.2 nanogram per mil. So this is really good for 10% human serum. This is an example of a PK assay antigen capture format. This is for adalimumab. TNF alpha is the capture antigen. We come in with the drug in human serum and close the complex. A type 3 is used for detection. The sensitivity is even higher here, so we can achieve a really high sensitivity with these type 3 antibodies as well. And if you think of a complex new molecule with maybe only one binding site, not a classical antibody, and maybe a Fab, you cannot form a bridge. You only have one binding arm. Type 3 or type 2 would be the solution for antibodies like ranibizumab, as shown in the upper and lower panel in the PK assay antigen capture format. Our fully human anti-idiotypic antibodies can be used in an anti-drug antibody ADA assay as a clonal control. Patients develop anti-drug antibodies which can interfere with the action of the drug or cause side effects, and a measurement of anti-drug antibody response is needed to assess safety of the drug. The challenge is that reference standards or controls are needed. Our suggestion for a better assay is to use the ELISA bridging assay pictured in the right panel, and we use anti-idiotypic antibodies as clonal controls. If you use the drug as capture and you use the labeled drug antibody as detection and an anti-idiotypic antibody to bridge these two, then this can be used as a calibrator or control for your ADA assay. Today, as a standard control, patient serum is used, but sera may contain viruses or other harmful substances and may be available in only limited amounts. Human monoclonal antibodies have advantages over polyclonal serum controls. Among them are standardization, unlimited supply, affinity determined, and defined isotype. And the data shows that the system works to find different affinity antibodies to mimic an immune response. This is an example of the ADA assay bridging format. This is for cetuximab. Type 1 antibodies are generated, and the secondary K-off rate screening, which I discussed previously, was used to move forward antibodies with different affinities. We took the 3 different affinity antibodies and used them in the bridging assay in the right panel. And as you can see, the sensitivity curve shift, which is exactly what you would expect. Antibodies we generate in the monovalent Fab format can be easily converted to full native antibodies with 2 arms to use in the ADA assay bridging format. Conversion can be to other isotypes as well as other IgG subclasses. This is another example of a high specificity antibody we generated. The challenge here was that we should generate antibodies to discriminate between a native and a truncated CXCL10 chemokine. There was one full-length version, amino acids 1 through 77, and there was a version, amino acids 3 through 77. It was truncated by only 2 amino acids. 2 projects were carried out. One project used the full-length protein as the antigen, and blocking was with the truncated protein. In the other project, the truncated protein was the antigen, and the blocking was with the full-length protein. We generated 2 antibodies. In panel A, the antibody specifically detected only the truncated protein, and in panel B, the antibody specifically detected the full-length protein. In this example, you can see that it is just not possible to generate these discriminating antibodies in an animal immunization campaign. This is another example of a high specificity antibody we generated. The challenge was that we should generate enzyme active site specific antibodies which actively inhibit enzyme activity. And this is for the active site of the West Nile virus proteinase complex. To do this, the wild-type protein was used as the antigen, and blocking was done with a mutant construct in which a histidine residue had been replaced by an alanine within the active site. See the structure in the upper panel. The substitution mutation only altered the active site, keeping all other putative epitopes of the wild-type enzyme intact. The rationale was that only phages that would bind to the immobilized wild-type proteinase were those that were specific to the wild-type active site. The ELISA data showed that 3 antibodies selected could bind the wild-type active site and not the H51A mutant in the top panel. Further, these 3 antibodies were shown to inhibit wild-type enzyme activity in the bottom panel. This is an example of cross-reactive antibodies. These antibodies were selected and recognized both the human and mouse homologue shown in this flow screening data in the left panel. The second antibody we generated recognizes the human protein only in the right panel. We can find matched pairs using guided selection core technology service. Here we have the capture antibody against IFN gamma. We used the complex of this capture antibody and IFN gamma and selected the detection antibody in the panning process and found a very nice, highly sensitive sandwich pair, which has a limit of detection of 2.9 picograms per mil in the Luminex assay. As a side note, we often provide the service of sandwich pair screening to customers. Antibodies generated in a project are tested against each other, all as capture and all as detection. Pairs that work together are identified, as in this example, using the Bioplex platform, where many antibodies can be tested at one time. We then take the 5 pairs with the best signal and do a titration ELISA to test the sensitivity in each case and rank the pairs for the customer. In summary, we discussed the proven success of the UCAL Platinum Library, and then we discuss our 4 services. The panning service delivers speed, only 8 weeks for a generation. The guided selection core technology service delivers high specificity antibodies. The secondary K-off rate screening service delivers high affinity antibodies. And the SpyTag technology service delivers quick formats and labeling. Lastly, we discuss examples of high specificity antibodies like anti-idiotypic antibodies and antibodies that can detect small molecular changes. Thank you for your attention. For more information on these topics, please check out our website at bioradantibodies.com.
Fantastic. Thanks so much, Renee, for a great presentation. All right, and with that, I will welcome Renee back for the Q&A. All right, first question here. So Renee, how fast can a project get started?
Yeah, great question. So we do 24 customer projects every other week. So theoretically, if we get your antigen in week 1, we can start the project the following Monday, week 2. So it's a very fast going project. Projects can start very quickly.
Excellent. Question here about the affinities of the antibody. So what kinds of affinities can the antibodies usually achieve?
Yeah, so in this standard generation process that I discussed in the webinar today, we can generate antibodies with affinities in the low nanomolar range and sometimes sub-nanomolar. And we also have another generation process, which I didn't discuss today, which is affinity maturation. So if that affinity is not good enough for you, we can do a single binder maturation. And routinely, we achieve 10-fold higher affinities, and improvements of more than 1,000-fold have also been reached. So yeah, that's as far as affinities, what we can achieve.
Great to hear. Thanks so much. It's a good question here. Is it possible to generate antibodies to things like DNA or carbohydrates?
Yeah, that's a great question. Those are historically difficult antigens to generate antibodies to, but we're more successful with those types of antigens because in the in vitro phage display process, we don't require an immune response, and we're also successful, for example, with self-antigens, which are also difficult targets.
Fantastic. So next question. So you talked a bit about using ELISA in your presentation, but what other platforms beyond ELISA can the antibodies be used on?
Yeah, good question. So ELISA, which I discussed in the webinar, but also other immunoassay platforms like MSD, Gyros, and Luminex, our antibodies are successful in those platforms too, as well as other assay technologies, Western blot, flow, IHC technologies, and we also have a secondary screen in the panning process for Western blot flow, which are employed in the same time as the KL franking, which I discussed. So yes, the antibodies are successful in other immunoassay platforms and in additional assay technologies.
Excellent, great answer. All right, so apart from bioanalytical reagents, do you also provide a service for the discovery of new biotherapeutic leads?
Yes, we do. We recently launched Pioneer Antibody Discovery Platform, and Pioneer is Bio-Rad's game-changing therapeutic antibody discovery platform that unlocks the discovery of clinical quality candidates in industry-leading timelines. And for example, the Pioneer library is 5 times bigger than our UCAL Platinum library. It makes great use of the library space in that germline sequences, which are proven, have been used, and other sequences have been removed. We also this time incorporated the SpyTag technology directly into the library construct, which accelerates the process greatly, and also helps the Fab folding because in the technology, the Fab is not fused to gene 3, as in our UCAL Platinum technology. And we also, in this Pioneer platform, we use the Trailblazer antibodies. And also we have a SpyLock technology, which is also on our website where you can screen very quickly by specific antibody candidates using the SpyTag technology.
Excellent. I think that's just about it for the questions, but maybe Renee, if somebody from the audience wants to come to speak with somebody from Bio-Rad, what conferences will you be at Bio-Rad this year?
Yeah, so we'll be at WRIB later on this month in Florida, and later on in the year we'll be at the European Bioanalysis Forum, PEGS, and also the Festival of Biologics in Europe, and also the Biotech Week Boston and Antibody Engineering and Therapy in the US. Those conferences will be attending.
Fantastic. All right. Well, Renee, it's really been a pleasure having you with us. Thanks so much for your insights today.
Yes, it's been a pleasure, and I really thank everyone for attending and for your attention.
Yeah, definitely. Big thanks to everybody for joining us. So in closing, we hope you enjoyed this Scientist.com Inside Scientific and Bio-Rad webinar, and we look forward to having you with us again soon. Have a great day, everyone.